Hello viewers, welcome back to my channel Knowledge Group, and today we'll discuss about the origin and evolution of seed habits. So you may understand more in less time. So first of all we'll discuss about what is the seed, and let's talk about this. So seeds consist of embryo, stored food, and seed coat. It's a complex and it's found in the vascular plants, and they have sexual reproduction, not the vegetative or asexual reproduction. And the vascular system means their xylem and phloem are diverse lineage. So their origin is about in the late Devonian times and 385 million years ago, like that. So seed is a ripened fertilized ovary, and seed is means defined as integument in the nascent megasporangium. Next is in this picture you can see that the cotyledons, the hypocotyledons radicle is known as embryo, and the seed coat that protect the seed and storage tissues means it protect the stored food or fruit and that we eat when they're ripe. And microphyls means that where the microspores are present, and here in the right, like in this picture, you can see the embryo plant body, their seed coat that protect the seed, and the cotyledons food for the plant, like it store the foods and so that we can eat after they're ripened. Next is the history. History means the earliest seed plants, progymnosperms, like the progymnosperms, then recent gymnosperms, and then the advanced gymnosperms. So in the progymnosperms emerged in the late Devonian period, and they show vegetative morphologies, mean they show vegetative structures like their physical appearance, and they did not have seeds, mean they did not have seeds or seed-like structure, means ovules or pre-ovules. And the species is Archaeopteryx is the first modern tree, and it produces spores rather than seeds, mean it produces spores, not the seeds. So it exhibit an advanced system of spore production called heterospory, advanced system that is known as the heterospory. So here is the diagram of the what is the Archaeopteryx species, and here is the first plant of the gymnosperm, progymnosperm. What is heterospory? What is heterospory? It's considered to be the prerequisite and to seed formation. And the two kinds of dissimilar spores means different in shape and different in size, and that is known as like their shape and the size is different but same species of different shape and size. It's known as heterospory. Heterospory is involved during the Devonian period of isospory. Isospory means same spores. So two types of spores, microspores and megaspore. Micro means small and megaspore mean large. So heterospory is reported in 9 genera, like 9 genera, and Saginella, Isoides, Stylites, Flurea, Regnilidium, and Marsilia, Sylvania, Ozolo, and Platysoma. So the next is the origin. Stewart and Rothwell proposed 6 evolutionary events, and the first one is evolution of heterospory, the retention and germination of megaspore within the megasporangium, mean megaspore is produced within the megasporangium, and the development of protective layer around the megasporangium, reduction of single functional megaspore per sporangium, development of embryo sac within the sporangium, and this modification of diastole and of megasporangium for pollen rupture. So the next is there is a 4 steps that you can see in this picture, and the first is that sporangium containing spores that are similar in size, means that the sporangium has similar size of spores. In the second diagram you can see the early evolution of heterospory, means size and shape are different but same species. So here is the containing many small microspores, and a megaspore in the B diagram. 2B diagram, megasporangium containing fewer large spores, mean large. Microspores mean small spores, and megaspores mean large spores. So reduction to 1 megaspore per megasporangium, like means that reduction to 1 megaspore and per like 1 megasporangium. And then next is integuments, means enclosure of megasporangium within integuments to form ovule. When fertilized, the ovule develop into seed and we get it. Next, the second point is that about their evolutionary trend is retention and termination of megaspore within the megasporangium. Like in some plants, megaspore is not allowed to escape from megasporangium immediately after its formation, and they permanently retained in megaspore. So the confines the megasporangium wall of the megaspore germinate to form the egg containing female gametophyte. The next is third point that you can see in the previous diagram, and it means that the some branches like structure of sporophytes surround the megasporangium and fused with the megasporangium to form the protective envelope or integument, that is this. Important change led to the evolution and formation of the ovule, which is not nothing but the integument in the adhesion of megasporangium. Synergial hypothesis. Synergial hypothesis you can see in the previous diagram about what is synergial hypothesis by Benson in 1904. The nuclear modification theory by Andrews in 1961, and the Loam concept about the Zimmermann in 1952. So in this diagram you can see the stages of the seed in the previous time. That is the first is the telome trusses comprises of fertile and sterile telomes that you can see in this diagram. But in the B diagram, the heterorangy and the heterospory, like means different in size you can see clearly. But in the C, megasporangium means large spore by sterile telomes, and D is the pre-ovule. That is okay. So the next is evolution of ovules. As I tell you, the evidence is supported in the field of telomes. Telomes is an integumentary lobe, and the integument with the microphylls is well developed by A. G. Long in 1966. That you can see, this is A, B, C, and D. And here is a species name that are shown in this diagram. Next, fourth is a reduction to single functional megaspore per sporangium. So each megaspore mother cell within a megasporangium used to produce 4 gametophytes, and there was a competition for space and food. So soon the earlier vascular plants adopt a new strategy. Only 1 megaspore is selected for further development, while the remaining 3 are aborted, as in the real life in humans. Like if there are 4 babies, then 1 is aborted, 1 survive and the other 3 aborted, or sometimes 2 aborted. So there are difference in their function and forms and needs or survival. Development of an embryo sac within a megasporangium. So a single healthy megaspore retained with the megasporangium germinate to form the egg containing female gametophyte, that is known as embryo sac. And what is embryo sac? Embryo sac is a female gametophyte of seed plants consisting of thin-walled sac within this nucleus, that containing the egg nucleus and other nuclei which give rise to form the endosperm or fertilization. The next is okay. So here we can see the difference between the gymnosperm seed plant and the angiosperm seed plant. So you can see there is a microphyll. Then there is an integument that surround the seed, and there's megaspore sites, means it has megaspores. In this diagram, our nucleus also present. So in this diagram you can see the style, ovary, and upper. Here is the stigma present. Here is the microsporophyll. There is an integument, there is a nucleus, it's a megaspore site, and fusillatus, and the ovary wall. So there is a huge difference between the gymnosperm seed and the angiosperm seed plants. So there is a lot of modification and changes after the time occurs. Next, sixth point is the distal end of megasporangium become modified by capturing the pollens. Pollination. What is pollination? Is a transfer of pollens from a seed plant containing the ovules, and if the pollen grain germinates, it gives rise to tube of the and these two are sperms into the female gametophyte. And if we eliminate, they can be dispersed by air or animals. Dispersed by air or animals. So after the if the pollen being trapped, so the large mega sporangium produce in the pollen and carry the male gametes, and the embryo sac fertilize and form the egg zygote and form the embryo is the last stage. So here is this diagram. You can see the A is the unfertilized ovule, B is the fertilized ovule, and the C is C. Means first stage is unfertilized, then fertilized, and then the third stage is that pollen entered through the microsporophyll and germinate a pollen tube, and the fourth is that germinate and release the sperm nucleus and fertilize with the egg nucleus and form the seed. So in this diagram you can see the immature female pine cone, and then you can see, and here is the egg present and the germinated pollen grains, and they fused and to form the seed, new like seed or embryo, that is diploid. These are the haploid, and in this stage, in the B stage, they are also haploid, but in this stage they become diploid in nature. And in this diagram you can see about their detailed diagram with the explanation is that it's a immature pine cone, then the megasporangium means के haploid है. And integuments and spore wall, you can see there is a spore wall, there is a megasporangium that is diploid in nature, and male gametophyte germinate with the pollen grain and fuse to form the seed or embryo. And in this this sperm nucleus, pollen tube, and egg nucleus, female gametophyte, that is megasporangium or microsporangium is the male. And here is the details about these fertilized, unfertilized, and gymnosperm seed. So thanks for watching the video. If you like, then please comment and share with your family and friends. And if you need in soft form, you can comment or mail in sciencestudyinfo@gmail.com. Thank you very much.